So dear students, today we are going to talk about RNA splicing. What is RNA splicing? As we know, when transcription occurs, it forms messenger RNA. Okay. In eukaryotes, pre-messenger RNA is formed first. Pre-messenger RNA is formed first. This pre-messenger RNA contains two types of sequences. One is called exon and the other is called intron. Exon and intron. This whole piece of messenger RNA is pre-mRNA. It contains two types of sequences. One is exon. This is exon. And it also contains intron. So what are exons? What are the introns? Introns are intervening sequences. These sequences are removed. And exons are joined together. And introns are removed. The whole process is called splicing. Okay. This intron sequence is removed and exons are, this is exon 1, 2. This exon 1 and exon 2 are ligated together to form a messenger RNA, which is called mature messenger RNA. Okay, this mature messenger RNA then codes for protein. Okay, dear students, what happens in eukaryotes? RNA splicing occurs. So, dear students, now we will see the sequences within the RNA determine where splicing occurs. Okay, here you can see this is an exon, this is an intron, this is an exon. In exon plus intron, there are 5 prime splice site towards the 5 end, and there is a branch point. This is called a branch point. Branch point contains adenosine, which lies close to the 3 end of the intron. This is the 3 end of the intron, and this is the 3 splice site. This site is 3 splice site, and this is the 5 end and 3 end of the pre-messenger RNA. Okay. The boundary at the 5 prime end of the intron is marked by 5 splice site. This is the boundary of intron. This is called a 5 splice site. Okay. The boundary at the 3 prime end of the intron is marked by 3 splice site. And this is 5 splice site. Branch point, which is found within the intron, usually close to its 3 end. This is more close to the 3 end, this branch point. All the 3 sites contain these sites, 5 splice site, branch point site, and this 3 prime splice site. They contain consensus sequences. What are consensus sequences? These are the sequences Consensus sequences are the sequences which are found in different species in same orientation and at same position. So dear students, what happens in RNA splicing? Basically, 2 reactions of transesterification occurs in RNA splicing. As we have studied about these the sequences within the RNA determine where splicing occurs. As we know, this site is called a 5' splice site, this is called a 3' splice site, and this is branch point. This branch point contains adenosine. At this adenosine of the this OH of the adenosine of branch point, it acts as a nucleophile. It acts as a nucleophile, and it attacks the phosphorylal group here of conserved G guanine in the 5' splice site. Okay, it attacks this, and it breaks the phosphodiester bond. The 5' free end of the intron is joined. The 5' free in this the 5' free end of the intron is joined to the branch point A adenosine within the branch point. It forms a lariat. This is one esterification reaction, and the second esterification reaction occurs when the free 3OH of the 5' exon becomes nucleophilic. It becomes nucleophilic and it attacks the phosphoryl group at the 3' splice site. Okay, then 5' exon and 3' exon, they join together to form spliced exon, and this comes out as an intron lariat. It forms the lariat structure. In whole of this reaction, there is no net gain of chemical bonds. As and if there is no net gain of chemical bonds, there hence ATP independent. There is no hydrolysis of ATP. ATP is not needed here in this reaction. Now in another lecture we will study about the machinery which is responsible for RNA splicing. Thank you.